In the past 3 years, we've done toxin wars, and we decided to make peace. We're not going to do toxin wars this year. We're going to do toxin science. So Rui is going to go first, and everybody knows everybody up here in this panel, and you've all worked at some point, it seems like, with Allergan for the most of you. But Rui is with David Montezzetti at Evolus. And come on up and let's hear about Evolus.
All right. Jeuveau is one of the newer toxins that was approved. It was approved by the FDA in 2019. It's also approved in Canada, all 31 countries in Europe, and just most recently Australia. It started as a 900 kilodalton complex that we licensed from South Korea. And when we looked at the phase 3 data from South Korea, to which they got approval, we were very intrigued. It was a head-to-head against Botox. And when we licensed the product, then we had the sobering conversation with Dave when we said, great product. It will never meet the criteria required by the FDA, ever. Manufacturing, nothing. So we then took that molecule and we designed it to our specifications, changed the manufacturing, and actually had an entire factory built to meet the specifications of the FDA. One of the things that we changed, for instance, was we went from freeze drying to vacuum drying. Technically, when you freeze dry, you create crystals, and a toxin is a delicate protein. So that freeze drying can actually denature your protein, and that can have immunogenicity issues. So we changed it technically, and we dried it with vacuum drying. So you change and you play with the algorithm, pressure, temperature, and how much time, and we're able to make the product without any crystals, no damage of the toxin. In fact, we don't need any overage. Clinically, what does that mean? Well, when we looked at immunogenicity, not only did we not make any neutralizing antibodies, just as important, we didn't make any general antibodies. Those are the ones that can give you hypersensitivity reactions. Clinical trials and approval, there were 3 phase 3 studies for the FDA approval. 2 were placebo-controlled, and 1 was actually a European-Canadian study with 540 patients. It was head-to-head against Botox, and both drugs obviously did really well, and that was published at ASJ, Fouad Nahai's paper. We also did long-term studies, and one thing that I think is intuitive to most is most of these studies are one treatment. What happens when you treat a patient for an entire year? What we found was 70 to 80% of the patients, depending on how you measure them, actually had improvements on their wrinkles at rest. Those are those wrinkles that are starting to become permanent. And it really attests to the fact that if you use the toxin for prolonged periods of time, the body has the ability to regenerate and actually repair. Most recently, another publication, that's actually interim analysis that we just shared, was we did what our other friends have done here also. We looked at a duration strategy. So when you take 20 units of a toxin or a Jeuveau, you get a duration. What happens when you use 40 units? And what we found was when we took it to 40 units in the glabellar region, we can actually get 26 weeks worth of duration. From a company perspective, there's been a lot of emphasis on we came to market. This is an underpenetrated market, so if you're a physician or in the investment community, it's an underpenetrated market, so our strategy was to try and build that market, and we really focused on trying to build the millennial up as a potential customer. And recently just published a paper with Patricia Ogilvy and myself, and we looked at toxin use in millennials versus that older generation. You'd think there'd be a lot of papers there, but there isn't. And what we found was we know that it works in the older population really well, and it worked even better in the millennials. You probably would imagine that because we know with age, toxin use comes down. What was surprising was how well it worked when you asked the subjects, millennials, what about subjective things like look in the mirror, global aesthetic improvement, subject satisfactions? To our surprise, the millennials did even better than the older generation. We saw scores of 100% in statistical superiority. Final comment, just as a company, we try to create a win-win situation. Most of our branding is targeted to millennials, and we're trying to build the brand of the company, but when we partner with customers and with doctors and HCPs, we're actually trying to build the brand also. I think that was right 4 minutes. Thank you.
Thank you. Thank you very much. Okay, Mitch, you're next. Let's hear about the 8,000-pound gorilla. Mitch Bryn is coming from AbbVie-Allergan.
Hi, everybody. It's good to see all of you. How many of you have worked at Allergan before? Patty says twice. I'm going to talk to you just for a couple of minutes about Botox as the foundational product. I think all of you know that. We've been on the market for over 30 years. We have over 50,000 exposures in our database, and we are basically a data-driven company. We pride ourselves on excellence and innovation, and actually laying the groundwork, which all of the other companies follow suit on. We also pride ourselves on our manufacturing, and that we are absolutely comfortable delivering what all of you use in your office. Our portfolio is quite rich. We are expanding our work. We're going into the lower face with platysma prominence and master prominence. We're also exploring the first novel toxin to the category, which is a rapid onset, short-lasting toxin, serotype type E. And we have several other assets that are in development at the current time, but we won't go into details on that for some time. I think the other thing is that we're very aware everyone's talking about duration, and I think many of you know that duration is really pharmacology 101 in the sense that if you increase the dose, you get a longer duration. All of us, probably all of us on podium, have demonstrated that with our assets. And I think that's something very important for people to realize. It's also very important to be able to read and understand the literature. There's several different variables you need to think about. What are the quality attributes of the study? What are the rating scales? Recognize that every company has their own proprietary facial wrinkle scale, and that applies for both lines at rest and lines with activation. So those are some of the things that I think it's important for you to know. That's pretty much it. I think our drug speaks for itself. I guess there's one other data point I'd share. We have over 4,000 publications that are peer-reviewed. And I think the other thing is in terms of the safety and efficacy profile, we've got 15 main indications on the market, and our dosing range is quite substantial. If you read our label, it goes from small units for things like strabismus and blepharospasm, all the way up to 400 units for some large muscle indications. So we're pretty confident in the quality attributes of our drug. Thank you.
How about any comments on onset of action?
Comments about onset of action. So back to the days when I was at Columbia University. So I've had a long, for those of you who don't know me, I've been using it since the early 1980s when it was called Oculinum. I actually named it. And then joined Allergan in 2001. In our original publications, we talked about fairly rapid onset of action of type A. I think Andy Blitzer and I published that a significant number of our patients, I forget the exact number, had onset or experienced an effect within the first day. The thing that you need to be aware of is onset and peak effect are very different, okay? Patients will often describe that they have onset within the first day or day and a half, but that's not peak effect. So the pharmacology will be different in terms of onset and peak effect depending upon not only the formulation but also the serotype. I also see that I still see patients at UC Irvine, and I see that across the board with our patients.
Thank you.
Happy to help.
Thanks. Okay, our next speaker, Andy Curry, is going to talk about Xeomin and the MERS experience.
Thank you so much. It's never a quiet mic. Well, thank you so much for me being able to come out and be able to speak about Xeomin. Before I jump into talking about the toxin itself, I kind of want to talk a little bit about the toxin landscape that we currently find ourselves in. And many of what the talks have gone on today have actually touched on a lot of this. And one thing that I want to mention that we have seen is that there seems to be a lack of awareness of the total amount of toxin that our aesthetic patients are actually receiving. And MERS is really trying to bring awareness to this because it has substantial implications for both our current and future patients. And we've actually seen in a global survey that 44% of aesthetic patients are also receiving toxin for therapeutic purposes. And this was not something that we had expected to see because then we started to think about, well, we're already using such high amounts in aesthetics. Well, then you start coupling therapeutics and you get an incredibly high amount. That same survey also found that a majority of healthcare providers don't tend to even ask about this aesthetic therapeutic crossover. It does not tend to come up in patient consultations, in the checkup, when the patient's normally coming in. So it's kind of maybe no one's really entirely aware of what this is. But we've also seen in the same survey that 2/3 of healthcare providers noted and they observed that it is taking more toxin dosing or more frequent toxin dosing to achieve the same treatment effect over time than what they have seen with previously. So I kind of say all of that to give an idea of, okay, well, what's the modern aesthetic patient really kind of experiencing? What does that look like when we think about the modern patient? Because we know that modern aesthetic patient is very different than the very first early patients that were part of the first aesthetic trials when it was 20 units in the glabella. And I mention all of that because as we know that the total toxin dose increases, the risk for lack of effect grows alongside of that. And so one aspect that we believe is so incredibly important about Xeomin is the fact that it was the first 150 kilodalton formulation that was intentionally designed without any unnecessary proteins. And we believe that is of incredible importance when it comes to the immunogenicity when thinking about the lifetime of treatment of our patients. And we've also seen, as noted, all the different companies have different dose equals duration trials where as you increase the dose, you get a longer duration. We've seen that with Xeomin, where we went from 20 units to even 100 units, 5 times the on-label dose in the glabella, and saw over 6 months of duration with the same safety profile. And that is just an example of one of the things that we think is so kind of incredibly important. And also, there was a variety of papers that came out over the past 2 or 3 years from the Hefter group out of Germany that looked at other aspects of immunogenicity, one of which they were able to kind of compile several different factors, and they found that when patients were being treated exclusively with Xeomin, elements such as dose use of the toxin, the long-term treatment of toxin, as well as the frequency of dosing, was not of immunogenic concern when the patients were exclusive to Xeomin. And that was something that we think is of incredible importance when we think about these patients that are not only starting now, many of these modern aesthetic patients are starting much earlier in life than we've ever seen before. They're going to be getting treated for longer periods of their life than ever before. And also, we already know that they're receiving higher doses of toxin. So with all of this together, MERS strongly believes that Xeomin should be considered a frontline toxin option concerning the lifetime of treatment of our aesthetic patients. I think that's all my main points. I don't know where my timer is. I don't want to go over. But that is just one of the largest aspects that we have found when looking between all the different, not only just the efficacy of the toxins, because all the toxins, they're all approved, they all work. No one's going to try to argue that they don't. But what MERS has really kind of pivoted is to think about the long term. How does the toxin work in 10 years, 20 years, 30 years? And not only then, because the therapeutic uses of the drug also start to come up, may become more applicable as time goes on for these patients. And that is an incredibly important part that we want to be mindful of this lifetime aesthetic journey of our patients.
Thank you.
Thank you.
Okay, next is Connor Gallagher, who's going to come up and tell us about the most recent entry from our Vance.
So thank you, Dr. Stevens, for the opportunity and this great panel of colleagues. So yes, let me introduce to you Daxxify. So Daxxify was approved by the US FDA on September 7th, 2022, representing the newest neurotoxin on the market in the US, and what we believe is the first innovation in neurotoxin products since Botox was first approved over 30 years ago. So let me tell you a little bit about some of the ways Daxxify is different. So Daxxify is different in terms of its clinical performance. So first of all, it was studied in a very large phase 3 clinical program. We had over 2,800 patients, over 4,400 treatments, and also one of the longest studies, our open label study, went out for 84 weeks. The product from a clinical perspective is also unique and different. So if we agree that getting patients from moderate to severe glabellar lines to none or mild is the clinically desirable endpoint. We showed in our pivotal trials that 98% of patients got to none or mild at peak efficacy at week 4. We also showed that 70% of those patients maintained none or mild status at 16 weeks or 4 months. Of those patients, the patients in the pivotal trials, 80% of those patients got to none at week 4, so no glabellar lines whatsoever. So if we take none or mild as the target endpoint for these trials, or the most clinically relevant endpoint, getting patients, and we want to understand how long patients remain in that none or mild state. When we did Kaplan and Meier analysis of our pivotal trial data, we found that the median time for patients to lose none or mild, so return to moderate to severe glabellar lines, was 24 weeks. Okay, so how do we achieve that clinically differentiated profile? Well, first, actually, let me talk about safety. So with that clinical profile, you might expect that there will be a different safety result. And in fact, the safety profile for Daxxify was very similar to what currently approved toxins. No new safety signals found, and the eyelid ptosis rate was 2%, which is right in line with what everybody else has seen for glabellar lines. So how do we get to that? So we get to this clinical profile, this unique clinical profile, with an innovative formulation. So Daxxify is uniquely formulated, a modern formulation, if you will, and at the foundation, or the quarterbacking that formulation, is our proprietary peptide. So we have what some people will refer to as a secret sauce. So it's a 35 amino acid, highly positively charged synthetic peptide that is a key component of our formulation. So with the highly positive charged peptide, we'll bind electrostatically to the core neurotoxin, and together, the peptide with other aspects of our formulation provide the clinical performance that you see with the product. And importantly, you get the clinical profile that we see with Daxxify, giving the same amount of core neurotoxin, so active ingredient, as in the typical 20-unit dose of Botox. Okay, so you're not giving more neurotoxin. So a couple of other tidbits around Daxxify that are relevant to both patients and clinicians. So first of all, no human serum albumin in the formulation. The product is manufactured without the use of any animal components in the manufacturing process. The product's room temperature stable prior to reconstitution, and after reconstitution, the product is stable for 72 hours. So that's 3 times longer than any of the other products that are currently on the market, and that's a testament to the formulation. The product's 100% manufactured in the US, and finally, based on our early experience after approval and after launch, we're not seeing that anybody needs to change injection technique to use Daxxify any way differently to the way they're using current neuromodulators.
How about the mixing? How about any differences in Daxxify versus mixing for Botox?
Right, so Daxxify comes in a 100-unit vial. We recommend reconstitution of that 100-unit vial with 1.2 cc's of normal saline. And when you do that, your 0.1 injection push gives you 8 units. So it's very similar in terms of what you would use from a push volume to a standard neuromodulator that you would give it at 20 units.
I have some other follow-up questions.
I'm happy to take follow-up questions after.
I know.
Thanks.
Okay, thank you very much. And Dr. Lin, come on up here. Ziming Lin is from Galderma, and she's going to talk a little bit about Dysport. Come on up.
Thank you.
Yeah, thank you.
So as we know that Galderma has Dysport, and Dysport is on U.S. market for over 10 years already. And Dysport has a high load of a toxin and performs very well clinically. So today I'm going to talk about QM1114, or retinol toxin A. It's our next generation of a neuromodulator there. So retinol toxin is our new toxin and has a very strong clinical performance there. And we've done clinical studies, phase 3 are all done. We have a strong and robust data. And also it's in liquid formulation, and it's ready to use. Some people thinking ready use, it's easy, right? Not only that. Ready to use, not only is easy, it also shows that you reach to the vial there, and the product comes with a homogeneous product from the manufacturer. And also, it gives you a peace of mind. You don't need to worry about the potential error made during the reconstitution there. So let's talk about the first of a clinical performance. We said that it has a strong clinical performance due to of the timelines. I give you 2 endpoints there. For example, for the patients, by the way, we treated 1,900 patients. So, for the GL-treated patients there, 76% of the patients at the end of month 6 still not go back to the baseline. For the LCL-treated patients, at end of the study, month 6 there, still not go back to the baseline. And for the GL-treated patients, 39% of the patients reported, I see the results on day 1. For the LCL-treated patients, 34% of them reported that I see the results on day 1. So no matter what is treated, over 1/3 of the patients are saying that I see results on day 1 there. So what underlines the results? We believe it's a manufacturing process. And Mitch talked about that a higher load toxin could give you a longer duration. We agree. And at the same time, we designed a manufacturing process that is gentle. It aimed to keep, preserve, and maintain the specific activity of the toxin A. This is going to be the only product that does not go through the precipitation process or dry freeze process, which are very harsh for toxin. We know toxin A is a very potent, product. While we talk about drugs, we talk milligram. While we talked about the toxin A, we talk about the nanogram and the picogram there. And so how we do that? At first, we go through the fermentation process, and we know during the fermentation process, it comes with a lot of impurities and proteases. And proteases will love to do is to destroy the toxin A. So we take advantage of this nature. And so while toxin A is protected by complex protein, and we use the filtration process, chromatography process, remove all the impurities, including proteases there. While we know toxin A is in a safe environment, in a liquid environment, we separate the complex protein from the toxin A and remove the toxin and remove the complex protein. This is how we make a pure liquid toxin without any complex protein, without any human or animal-derived excipients there, and performs very well clinically. That is our ralobotulinum toxin, QM1114. Thank you.
You all have microphones, I trust. Dr. Lin, I'm curious about this toxin. Tell me again the name of it. What are you calling it again?
It's a QM1114 during the development process. USAM name is a ralobotulinum toxin, and the brand name, and we will announce later.
Okay, fair enough.
Sneaky.
I remember when Fleming first talked about it on the technology of beauty, and he came over. I'm still a little confused. Is this a dose-related phenomena that you're talking about? You said you strip all the proteins out. Do you add anything, such as the peptides that are added here when Revance does Daxxify? Or
No. So what we do is we focus on toxin and toxin alone. So we put enough ideal load of a toxin on this product, and we designed a manufacturing process aimed at keeping the activity of the toxin. So during the process, it's a process, and like I said, this is the only product that will not have the precipitation process and the dry freeze process, which are harsh for toxin molecules.
And the studies that you're doing now, you have preloaded syringes, correct? And that's what you're going to come to market with?
It's a vial.
Okay, pre-mixed in a vial. So the practitioner will still draw it up?
It's a draw up and a dose, yes.
Okay, but it's pre-mixed, that's the point.
It
and
is.
then how is it stored?
It's a cold chain product.
Okay, thank you. Connor, where are you with therapeutic?
Therapeutics?
We've only been talking about aesthetic, but I'm curious. There are therapeutic indications for some of the other ones here. Where are you at with therapeutic?
So we've completed phase 3 in cervical dystonia, and that's under review at the FDA at the moment, and we have a PDUFA date, so we anticipate from the FDA in August for the cervical dystonia indication. We've done a phase 2 study in upper limb spasticity, and we're looking at potentially moving forward with that, and we're also considering going into the next indication might be migraine.
Can you give us any indication of what the outcomes were in the phase 3?
So the phase 3 for cervical dystonia showed a very robust clinical response. So a high degree of efficacy. Duration in the 2 doses that we studied was in the 20 to 24-week range. Importantly, from a side effect profile, in cervical dystonia, as many people know, just like eyelid ptosis is the sentinel adverse event in glabellar lines, the sentinel adverse event in cervical dystonia is dysphagia. And so our dysphagia rate was super low. It was in the 3 to 4% range in our pivotal trial, which compares to 15 to 20% for other toxins in their pivotal trials that are currently on the market.
Are you doing any other concurrent therapeutic trials? Are you starting down and looking for other indications in the therapeutic arena?
So what I've mentioned is where we're at now.
Okay.
We'll make further decisions later.
Okay, good. How about others, therapeutics? Obviously, I know Botox is. Evolus, have you even tried the therapeutic?
No, Evolus's position is pretty clear. It's aesthetic purely. The entire
right.
company is based on private pay,
Right.
non-reimbursed.
Okay. And MERS with Zim?
Yes, well, with how the business is split, so that MERS Aesthetics is pretty much its own separate business, so it's a shared product, but MERS Therapeutics kind of operating out of Germany. I have to poke in to ask them what it is that they're actually doing. But I know recently they've had chronic sialorrhea as one of the therapeutic indications. I won't speak to what I think that they have on the precipice of other things that they're wanting to kind of explore.
It's sialorrhea.
Sialorrhea, yes.
That's right.
I'd like to know, show of hands, how many you think that the reason that Daxxify lasts longer is the dose, the number of units? Go ahead, don't be shy. 1, 2, 3. How about you, Dr. Lin? You see
I do not know.
excuse me?
I don't know, so I cannot,
know.
yeah.
Okay, that's obviously one of the big debates on duration, yet we just heard Connor say it's the same number of units or the same weight of the toxin, if you will. So why don't we talk about that back and forth? You take the lead, because I've already heard you.
So I should take the lead?
Yes, please,
Oh, okay.
go right ahead.
So actually, one point about development. So we're in development also for interstitial cystitis, essential tremor, and we have another program in postoperative atrial fibrillation. So we're going into some pretty serious medical conditions. So there's a lot of debate about units and a lot of debate about mass. So a couple of things to realize. First principle is that every company has their own reference standard and they calibrate their units differently by a proprietary reference standard. Okay, so it's the FDA that put into all of our labels that the units are interchangeable. And the first time that went into the FDA label was in December of 2000 when MyoBlock was approved. And they're very passionate about that. It's in all of our labels. It's also in several other places in our labels. So there are a couple of publications about mass. One comes from Dysport, one comes from Mertz. I think Jürgen Fravert is on it. And they do some calculations and they translate that into units. And if you go through all of the calculations and then you say, okay, we're on the market for 20 units for glabellar lines, and you take the mass and you run the calculations, and I've got a slide that shows this, it comes out to, if you believe that, then for Xeomin you're using either 8 or 11 units on market, okay? But in reality, it's labeled for 20 units. So you cannot take those mass calculations, draw a straight line to units, and then draw a straight line into the indications for which it's labeled for. So a lot of that just doesn't add up. And I think at the end of the day, when you're treating a patient, you're treating biology. So you're treating with biological units. And each company has their own units, their own label, their own calibration. And that's what we all train people to use.
Okay, we're getting close to wrap-up time. I'm going to jump back to Connor and let him counter.
Okay. because I have a counter for the two, a real simple one.
Okay, go ahead.
Yeah,
You
it's
go
really
first.
simple. The whole argument is Brevance has 0.18 nanograms, Botox has 0.18, and supposedly we have 0.12 nanograms in a vial. The simple answer, I agree with all the points you made, Bitch, those numbers are impossible. I'll repeat it. Those numbers are impossible. Every vial, every lot will have variability in the number of nanograms. Number two, there isn't a machine in the world that can fill a vial to that kind of specification. It doesn't exist. That's why there's a release specification. Number three, Brevance actually never measured. If you look at the references, they've actually never done those measurements. So those are my quick 3 points.
Okay, thank you. Connor, the floor is yours. Wrap it up.
Okay, happy to talk
I know
about
you
that.
feel outnumbered here, but go for it, buddy.
Yeah, that's okay. I'm used to it.
You've worked with all these guys.
So first thing, I 100% agree with Mitchell in the sense that units are not interchangeable. All the labels say that, and there's good reason for that, right? Units are a useful measure for dosing within a toxin. So for patients on Botox or on Dysport or on Jeuveau, dosing for how those products will be used. But it is not a basis for comparison between toxins, as the labels have said. So most other medicines are dosed on the basis of mass, okay? So ibuprofen, biologics, other biologics are dosed on the basis of mass. Mass is a universal measurement, and you can quantify the mass of core neurotoxin by ELISA in the vials. And so, yes, these are dosed, but initially filled by units by each company. They're proprietary units. But you can measure the amount of neurotoxin in each of the vials. And so the values that Rui mentioned, 0.18 nanograms, are the nanograms as being reported from others in the literature, in the scientific literature, peer-reviewed, for the mass of core neurotoxin in the 20-unit dose of Botox, for example. And so mass is mass, nanograms are nanograms. You can quantify it, you can measure it, and people publish it in peer-reviewed journals. When we quantify the mass of core neurotoxin in Daxxify, in our 40-unit dose, which is our approved dose, we have approximately 0.18 nanograms of core neurotoxin. So you can quantify it, you can measure it, and you can report it. And while we don't dose on that basis, it is interesting, and it's an important way for us to understand the differences in formulation. Because at the end of the day, people can dose whatever they want. But what we're suggesting here is that when you give a similar amount of core neurotoxin in Daxxify versus, say, Botox, you get a differentiated clinical response. That is driven by the formulation in Daxxify, which is unique and supported by all the components of the formulation.
Well, I don't think we're ever going to agree. And
but
we've
can I ask
gone
a
over.
technical question?
No, we've gone over.
Okay.
We've gone over 2 minutes. I have to, we could do this for the next day, I know that. So I'm going to thank you all for joining us. I hope you come back next year. Maybe we can figure out the dosing and the units and on and on and on. I don't think we're going to agree. So thank you very much.